everyone. Thank you so much for coming, for being here. You are all my family, and I can't thank you enough for your support. It really touches my heart. I'm a little touched by the fact that in this short time we were able to put this all together during the pandemic. It was difficult, but my wonderful assistant, Caitlin, was able to take charge, and she did a magnificent, magnificent job. That's right. And Dr. Israel, you're an angel. People don't really know you, but I can tell them that your heart and your caring and helping makes such a difference for cancer. I take my hat off to you. And Dr. Raz, thank you for your amazing knowledge and ability to teach us so that we could learn and have quality for all people. I want to tell you that this has been the most difficult time. I'm a little teary-eyed tonight, but this has been the most difficult year we have all experienced in this horrific pandemic. But I have to tell you one thing. It did teach us the importance of taking time to learn and lessons, and we've changed a lot of thinking. A lot of families are thinking differently because of the pandemic. Their whole world, science researchers have focused on a cure, that they found a solution. That should encourage all of us. They all worked so hard. In 7 months, they were able to find a solution to help us survive, to live. Cancer today needs that kind of research, like the pandemic. We need to join hands, and throughout history, we have solved so many diseases because of research. In fact, in the cells of the diseases lies the answer we are looking for. We have made massive degrees of finding many cures, but there are still so many we have not been able to find. And we need your help. With your generosity and your help, there will be less cancer in this world, and our children will grow up knowing that they will survive and they will be healthy. And it's only through your funding and your attention to cancer we will be able to do this. I'm asking all of you, join hands, come together. We are a family. We need your energy. We need your time. We need your money. But it's not a lot that we're asking for, because it's giving us time for us to live longer and have more fun in this world. So I want to thank all of you for being here tonight. You have no idea of what it means to our Israel Cancer Research. Without you, this would not be possible. I take my hat off to all of you for your giving and caring, and God bless you. At this time, I want to introduce my wonderful doctor that's been with Israel Cancer Research for 5 years now. Is that correct?
Sure.
5 years?
Even longer, actually.
Even longer. He has dedicated his whole days and weeks and months, never stopping, to find us and help to get money so we continue with our grants. Our grants are the only way that we support science. And we have many grants that we need to support. And Dr. Mark, God bless you and continue with us, because we couldn't do it. We would not be able to do it without you. Thank you.
Well, Beverly, and thank you very much. It really is supporters like you that make all of this possible. And I'm so pleased that you and your friends are so engaged with ICRF. As Beverly indicated, I am the past executive director at ICRF. I'm a cancer scientist and an oncologist. And before coming to the Israel Cancer Research Fund, I was director of the Cancer Center at Dartmouth Medical School. For those of you who aren't too familiar with ICRF, I'll just say very briefly that it was founded in 1975 by a group of American and Canadian medical researchers and oncologists and lay people who were committed to the growth and development of Israel and to combating the worldwide scourge of cancer. And these visionaries were determined to harness Israel's educational and scientific resources in the fight against cancer while supporting the state of Israel. And their solution was to provide funds for young Israeli MDs and PhDs to conduct research in Israel, and that solution worked. In addition to ICRF's many achievements and medical breakthroughs, ICRF supported scientists. The organization now operates chapters in half a dozen cities in 3 different countries. And with its international headquarters in New York City, we have a remarkable cadre of hundreds of committed lay donors to our effort. I think you'll be surprised to know that year in, year out, Israeli Cancer Research Fund provides support for over 70 different cancer research projects at any given time. That's more than any other non-governmental agency. And over the years, these funds have supported research at all of Israel's major biomedical research centers. And in the 45 years that ICRF has been active, we've raised and distributed over $77.5 million. $77 million. And our people that we have support, scientists that we support, have won prestigious awards, including the Nobel Prize, the Israel Prize, the Lasker Award. It's just been an incredible ride. Now, before I introduce Dr. Dan Raz to tell you about his amazing research, I just wanted to touch on a couple of other really exciting things that are coming out of Israel now. And I'm thinking of Dr. Ben David's work at Hebrew University, where he focuses on a particular trait of cancer cells called aneuploidy, where there's a abnormal number of genes, of chromosomes in cancer cells. Now, this only occurs in cancer cells. So if we could develop a treatment that targets this, we wouldn't have any side effects of these treatments because they would be targeting something that only occurs in cancer cells. And there's other work, for instance, by Professor Ben Eliyahu at Tel Aviv University that focuses on how stress, and believe me, during COVID, stress is a huge problem, how stress on our immune system allows for the development of cancer metastasis, allows for cancer to spread throughout the body. And his work has yielded a promising treatment that we believe holds great hope to reduce the risk of metastasis that occur in this case during following surgery for colon cancer. And there's now a worldwide clinical study ongoing to test these interventions. And I'll just mention one more that comes to mind, the work of Irit Ben-Aran at Rambam, where she is the director of the Division of Oncology. You may know that Rambam is the largest cancer center in northern Israel, and she's studying how chemotherapy damages blood vessels and leads to fertility problems and vascular disease in survivors. And so we hope that her current ICRF funded research will improve the quality of life for survivors, and by diminishing the amount of vascular disease and also enhancing chances for a successful pregnancy in these women affected by this terrible side effect. And I could go on and talk about other things. We have a magnificent program sponsored by a variety of donors, the Jackie and Bruce Barron Scholar Program, that focuses primarily on symptom relief and palliative care to improve the quality of the life of cancer patients. So there's a lot going on in Israel, and it'll change the way we treat cancer in the future. I have no doubt about that. But one of the most exciting programs going on at ICRF is one that provides funding for Israeli investigators and American investigators to interact. And I want to introduce now Dr. Dan Raz, who collaborates actively with Professor Ravid Straussman at the Weizmann Institute, conducting what I believe is uniquely exciting research funded by dollars that folks like you contribute. Dr. Raz is at the City of Hope Hospital, where of the 70 or so grants that we're funding this year that I mentioned, there are now 5 different interactive projects between City of Hope scientists and Israeli scientists that are funded through the ICRF. Dr. Raz is a particularly accomplished individual. He's a thoracic surgeon, a chest surgeon, and he is an active cancer scientist who completed his clinical and research training at the University of California in San Francisco and the Massachusetts General Hospital. He now is the co-director of the lung cancer oncology program at City of Hope, the director of the Baum Family Thoracic Oncology Laboratory, and he himself founded the lung cancer screening and tobacco cessation programs that are so successful at City of Hope. And that goes back quite a ways, to 2012 or so. Now, lung cancer, which is a particular interest of Dr. Raz's, is amongst the most deadly of the commonly occurring cancers. And the major focus of his lab has been to study the resistance of lung cancer to anti-cancer therapies, to chemotherapy. You can imagine that if tumor cells responded to cancer treatment perfectly, everyone would be cured. But unfortunately, many individuals with lung cancer have tumors that are resistant to all of the treatments that we currently have available. And since 2016, Dr. Raz has been working on a collaborative project with Israeli investigators to pursue a most unexpected finding. And I'm just going to tell you what that finding is and then turn this over to Dr. Raz. Now, all of you know that inside the human body, below our skin, the tissues are sterile. I mean, there's no bacteria or infection. And if there were bacteria in your body, you would be sick with an infection. But it turns out that within cancer cells, there are bacteria inside the cells, not in normal tissues, not under any normal circumstances, but in cancer, the bacteria sneak from between the cells and in the blood, and they get into tumor cells. And using support from ICRF, Dr. Raz has found that there are specific bacteria that might be leading to the resistance of cancer cells to specific lung cancer treatments, to resistance to the drugs we have now to treat lung cancer. And I've asked Dr. Raz to talk about that a bit tonight. And then my plan is to chat with him about the implications of his work. And then if any of you use the chat function at the bottom of your screen to send me questions, we'll try to work together, Dr. Raz and I, to answer those questions. And then we'll wrap this up. So welcome, Dr. Raz, and thank you so much for being here with us tonight.
Thank you. Thank you, Dr. Israel, and thank you, Beverly, for putting this together. As Dr. Israel mentioned, I'm a thoracic surgeon at City of Hope, and I've had the great fortune to be supported by ICRF in my collaboration with Dr. Ravid Straussman in Israel at the Weizmann Institute. I'm just going to share. Dr. Straussman is a brilliant scientist. I don't think brilliant does him justice, but his area of expertise is the interaction between bacteria and cancer and how that affects sensitivity to different types of cancer treatment. So when we first met, he had just completed really groundbreaking work where he was really focused on pancreatic cancer at the time. And he identified specific types of bacteria living in pancreatic cancers that actually caused resistance to one of the most commonly used chemotherapy agents, gemcitabine, for pancreatic cancer. And to be honest, when he contacted me, and this all came together because of ICRF, when he contacted me, it almost sounded too good to be true. And this was later published in Science, which is a very prestigious journal. And he approached me asking, can we start to look at things in lung cancer and see if we can find similar types of bacteria that cause sensitivity or resistance to specific types of lung cancer treatments. So what we found was, indeed, there are bacteria inside of lung cancers. There's actually bacteria in all different types of lung cancer that Dr. Straussman and his collaborators have since examined. And these bacteria are all different. So lung cancer patients tend to have specific types of bacteria. Breast cancer patients tend to have other types of bacteria. And the really neat thing about lung cancer is that there's certain bacteria that are found, for example, in smokers, people who smoke or used to smoke. And those are very different than the bacteria that are found in people who develop lung cancer who've never smoked. Now, we're not at this point quite sure what that means, but this was, a very fascinating observation. And our data, along with data in other cancers, was published in Science last year. And these are some pictures showing bacteria. So for example, on the bottom left rectangle, where you see the red and the blue arrows and the pink thing, that's actually bacteria within a cancer cell. So the next step was we wanted to figure out if certain types of bacteria that we identified in lung cancers led to resistance of specific types of lung cancer therapies that we use. Now, 20 years ago, everyone who got lung cancer, when we used chemotherapy, would get the standard chemotherapy. But now there are targeted therapies. So those are medications that attack cancer cells that have specific genetic changes. And we have immunotherapy, where these are drugs that harness the body's own immune system to attack lung cancer. And these targeted therapies and immunotherapies have been a huge advance for lung cancer patients and resulted in really marked improvements in survival, even in patients, where the cancer has metastasized or spread. So our big first observation was that the most commonly used targeted therapy, so this is a class of medications called EGFR, tyrosine kinase inhibitors. So you can see in this top bar, something called osimertinib is now the most commonly used medication for EGFR mutated lung cancer. It's found in about 20% of lung cancer cases. Actually, in Southern California, it's closer to 30% because there's so many never smokers who get lung cancer. And what we found is that when you add the broth of a particular bacteria that we identified in lung cancers, it actually leads to resistance to that medication. So this is the one thing I want to show you that's important. So you can see the top bar all the way, the first square, those are all cancer cells that are green, that are lit up. This is a special kind of marker that identifies the cancer cells. The next square is where we add the drug, osimertinib. And you can see most of the cancer cells are now killed. There's not as many green dots. The next square is just adding the broth from this bacteria. So it doesn't actually contain the bacteria, but the bacteria were in this broth and we're just adding the broth to the cells, nothing happens. And when we add the medication with the broth, you can see that there's a little bit fewer green cells, but nothing like the medication alone. So there is resistance from whatever is in this broth. And this is what we've been working on. And this is just a cartoon to show you how more specialized lung cancer treatment has become. We used to just categorize lung cancer cells based on how they looked under the microscope. Adenocarcinoma, squamous cell carcinoma, those are the two biggest categories, or small cell lung cancer as well. And now it's standard to get genetic profiling where there are certain changes, genetic changes, and like I mentioned, EGFR is one of the most common ones that really affect in a huge way how patients are treated and how they respond and how they cure. But unfortunately, despite this big advance, all of these patients eventually develop resistance. Some are really lucky and they don't develop resistance for 5 years or even 10 years, but that's pretty uncommon. Most of the times, people respond to the treatment for maybe 2 years and then start to develop resistance. And there are some people who develop resistance really fast. And we really don't understand why that is, why some people develop resistance really fast and other people can live 5 or 10 years. And if there was some way that we could, outsmart the cancer and overcome that resistance, wouldn't it be amazing if, a diagnosis of metastatic lung cancer, someone could survive 10 years or even longer. And so what we did, and the details of this slide are not that important, but what we did was we took this bacterial broth, and the first thing we did was we added it to cancer cells that had this change, this mutation, and we looked at all the genes. So we did genome-wide analysis at all the genes and looked at how they changed in expression. And you can see these are some top genes that changed in response to adding that bacterial broth. And you can see in the square on the left, there's one called CA9 that was a marked increase. So these are the kinds of things we're looking for. These are the kinds of gene changes that give us clues to start to understand how this broth affects the response to lung cancer therapy, in this case, osimertinib. And these are just other genes that had, pretty significant changes. The actual names, that's just more, so I can show you what we're doing. And then the next thing that we did was we separated all of the different proteins in the broth into different sizes. So we're able to sort out all the proteins to different sizes to try to tease out what are the proteins in this broth that lead to resistance. And then we put it through a machine called a mass spectrometer that identifies the specific proteins or give us clues. And ultimately, these were the top proteins that we identified in the broth that led to resistance. And so right now we're in the stage of really trying to validate this and trying to put everything together and understand how this works. And we're also testing other bacteria that are related to this one specific bacteria that we found to see if they also lead to resistance. So I'm gonna I don't want to talk too much about this, but I want to just talk about the next step. So what does this mean? How do we turn this discovery into something that helps people? So right now we're still in the phase of understanding the mechanism. Maybe that means we can target specific bacteria. Maybe it means that we can develop specific drugs based on how those bacteria work to overcome resistance. Maybe it means people can take something like a probiotic and it prevents cancer? We don't know. So these are the things that we're trying to understand and learn. In the future, and now we're just starting to plan further experiments, we're trying to capitalize on this observation that bacteria in smokers versus never smokers are different. And we know that the type of lung cancer that smokers get and the type of bacteria that never smokers get are often very different. And the EGFR mutated lung cancer is one example of a type of lung cancer that's much more common in people who've never smoked or have a very light smoking history. And then the last really interesting thing that is, again, Ravid is so brilliant and working on, and he's been working on this in melanoma, is the role of the bacteria in the response to immunotherapy in lung cancer. And that's another big project that we're hoping to get underway. Ravid actually just published something in Nature, another really top prestigious journal, about just this in melanoma, about the kind of potential role of bacteria in melanoma cancers and the response to immunotherapy. And melanoma is another cancer where immunotherapy is very commonly used. So I'll end here. I want to say thank you. And I also just want to offer my contact information in case, unfortunately, there are people get cancer. If you ever need any local LA advice or any way I can connect you with people at City of Hope, feel free to contact me.
Dan, thank you so much. That was remarkable, and your generosity in offering contact information is just a refreshing delight. You mentioned, of course, that this is an issue in melanoma as well, but I'm curious to know that the general gist of this is that the bacteria can affect immune therapy or chemotherapy or targeted therapy, but is it, if you have a particular bacteria that affects lung cancer in a particular way, is that same bacteria affecting other tumors like melanoma in the same way?
Well, that's a really good question. And as I was mentioning, there's actually different types of bacteria and different types of cancer. So on the one hand, it may be that certain bacteria, for example, the pancreatic cancer story, that bacteria actually makes an enzyme that degrades that particular type of chemotherapy. But when you talk about immunotherapy, having bacteria inside of a cancer may be more of a marker that immune cells cannot enter that cancer for some reason. So perhaps the bacteria is more of a kind of a clue that the immune system isn't activated against that cancer. So there's a lot of possibilities, and this is really, and again, this is all the props to Dr. Straussman. This is really paradigm shifting, and I think there's going to be a lot of interesting things that come out of this.
Dan, an interesting question that was sent to me is that you suggested that there, in any given type of cancer, there were many different kinds of bacteria in the individual cancer cells. Do you think the effect on drug resistance is the result of a single type of bacteria or a particular collection of bacteria?
Yeah, that's a good question. Just to clarify, so we haven't seen many patients where there's multiple different types of bacteria within one cancer. So typically one patient will identify a few bacteria of the same type. But what I meant to say was that in lung cancers, there's a variety of bacteria, and some patients will have one of several different types, whereas in breast cancer patients, they have a completely different set of type of bacteria. So there seem to be conserved. It's not like in one person, they might have the same bacteria in a lung cancer versus a breast cancer, and we really haven't seen too much of multiple bacteria in a particular cancer. I'm already forgetting what you were asking as the main question, though.
No, is it a single type of bacteria that's giving this effect, or do you need multiple different bacteria in a given tumor, multiple different types of bacteria in order to get the resistance to chemotherapy?
It seems like it's specific types of bacteria. And one of the things that we're trying to look at, for example, in this story, we found a very specific bacteria, but we're starting to look just now at other bacteria that are similar. So they're maybe in the same genus or just related, evolutionary relationship to that particular bacteria, to see, because oftentimes, certain enzymes or proteins that are secreted by a bacteria are secreted in other bacteria that are similar.
It's so interesting. Another question was, how is it that the bacteria actually get into the cells? We usually think of bacteria as living between cells or in the soup of tissues, whereas viruses sometimes do get into cells. How do bacteria get into cells? And do these tend to be gram-positive or gram-negative bacteria?
Great question. I think for the lungs, it's probably a little bit more straightforward. When we breathe, we have bacteria in our mouth that, can get into our airway. I think things like that, things like melanoma, where it's on the skin, so there's contact with bacteria. I think those are more straightforward in terms of how do the bacteria get there. How do they get there in pancreatic cancer or, breast cancer? I don't think we really have that answer. I think that's a little bit more curious to me. And then with regards to what types of bacteria, we see different types of bacteria. The specific type of bacteria that, for example, in our EGFR resistance is a gram-negative bacteria. It's called Chrysobacterium, that's the genus. And it's not a bacteria that causes infection. It's actually found in soil most commonly. And it's been reported to cause infection in people who are immunocompromised, but it's not, something that causes pneumonia typically.
Mm-hmm. Dan, I want to switch the topic a little bit before we end this evening, and that is that I can imagine that in the future, when we do a biopsy of a lung cancer, we'll want to know what bacteria are in it, because it may define how we treat it, that particular tumor. And so more precise diagnosis is one aspect that could come out of this work. But early diagnosis is oftentimes the result of cancer screening, of some sort of approach to looking at people who think they're well, looking for very early disease and that sort of thing. Are there active programs in cancer screening for lung cancer, and does this work have any implications for that?
I'm so glad you asked that, because as you know, lung cancer screening is a big passion of mine. And it's a passion of mine because lung cancer is by far the most common cause of cancer death. But when we find lung cancer early, we can actually cure lung cancer. The problem is that even though lung cancer screening, which is a very simple test, it's done with a CT scan, a low dose radiation CT scan, and it's recommended in people who smoke or used to smoke, who are between the ages of 50 and 80. So even though that's available, that's recommended by all of the panels that recommend cancer screening, it's fully covered by insurance. And despite that, very, very few people who are eligible for screening actually get screened. And to me, that's a real tragedy, because we could really save a lot of lives with lung cancer screening. So I'm really glad that you brought that up. And the question is also, does this work with the bacteria have any implication for early detection? And I think yes. I mean, I think just the finding that there's different bacteria in smokers versus never smokers, and the fact that patients who are never smokers who get lung cancer have very different types of lung cancer than people who smoke, by and large. Not always, but quite often. And these never smoking cancers are particularly common in women, in people who are East Asian or Southeast Asian heritage. And we really don't understand why that is. People have tried to look for genetic changes and really haven't, in kind of germline genetic changes, and haven't really found anything. And so we really want to do some digging into whether these different types of bacteria in people who smoke versus never smoked have anything to do with how lung cancer develops.
Well, that's great. I'd like to actually end on that note, because I would agree that it's a missed opportunity for people who are eligible for screening who don't get screening. But there was one question that I don't think we got to that I'm curious about as well. Is it known how the bacteria actually get into the tumor cells?
No, we don't know. And actually, we don't just find it in the tumor cells. We actually, so often, most of the time when we have cancer, and specifically lung cancer, there are all kinds of immune cells that are also there. And Dr. Straussman has also found bacteria actually inside of the immune cells also. Now, whether that's the immune cells carrying the bacteria there, that's a possibility. Whether it's the immune cells there to kind of clean up and try to get rid of the bacteria seems more likely. But again, there's a lot that we don't know about how these bacteria actually get there, in addition to what their role is.
Dan, thanks so much. These are amazing stories of Israeli science and collaborative science between Israeli scientists and American scientists, especially here at the City of Hope. I'm so proud of ICRF to be associated with work of this remarkable quality, and it's really game-changing, innovative impact. I want to thank all of you who are here, but I especially want to thank Beverly and Robert for all that they do for ICRF, their support of an ICRF grant in the past, a remarkable philanthropic contribution, and Beverly and Robert, your engagement tonight and your making this possible through your home and so forth has really just been great. I hope very much that our remarkable audience understands that all of this is made possible by the funds that they donate. And just this past week, we reviewed projects for funding for next year, and there were over 150, I think 170 odd projects that were reviewed. The science was amazing, and I'm absolutely certain that there will be grants that our 60 or so review scientists found worthy of funding, but for which we will not have funding. And who knows, maybe one of those grants will have been the grant that could have had a big impact on curing some particular type of cancer. So we don't want to miss those opportunities. We want to encourage you to support ICRF so that we can support Israeli cancer science. And with that, once again, thank you, Dr. Raz, Beverly, and Robert, thank you so much. And with that, I'm going to end the webinar and sign off. Thank you all very much. Good night, be safe, stay shuttered. We still got a ways to go here. Take care, everyone. And thank you very much.
Dr. Israel,
Yes.
Dr. Israel, I wanted to end that. I want to thank everyone who is here tonight and to tell all of you that I, at normal times, would have it at our hotel, and we'd be having drinks and dinner, cocktail reception. And so I owe each and every one of you at our third event, because we will be doing a 3-part series. Tonight is about lung. What is the second part? and the third part that we are offering?
I think we're still working on the program, Beverly, but we're going to try and identify tumors and advances that will be of interest to the greatest number of people.
Okay, and so we will have our next series approximately in 2 months, would you say?
I
Be
think
ready for
that's-
that?
Yeah, we can aim for
Okay.
2 months.
Okay, in 2 months, all right. So mark your calendars, we'll send you out notices. And then we are also at the third part, going to be doing it at our hotel for cocktails and dinner. So please stay with us. We have so much more to share with you about cancer research. This is only our beginning. And I want to thank each and every one of you. And Dr. Israel, God bless you, Dr. Raz, for all that you have done. And we will look forward to all being together. God bless. Thank you. And thank you, everyone in the audience. God bless all of us and your families.
And thank you so much.
Thank you.
Okay, Beverly, thank you so much.
Okay.
All right. Good night, everyone.
Good night. Thank you.